Welcome everyone to today's OTS webinar with Dr. Stanley Crooke. It's an honor to have you here, and he will tell us about the n-Lorem Foundation, who's doing really great work for the N=1 trials and treatments. As usual, the presentation will be recorded and will be available from the OTS website in a few days, so please be a little bit patient. All the attendees will be muted, and please put your questions in the chat box, and Dr. Crooke will answer them at the end. Thank you very much. Over to you.
Thank you, and thank you for joining me. Thanks also to the leadership of OTS for giving me the opportunity to introduce n-Lorem to you. n-Lorem is a charitable foundation that I initiated just about a year ago. And our mission at n-Lorem is to take advantage of the technology that we created at Ionis to bring experimental ASO treatments to patients with unique, ultra-rare mutations. And to do that for free for life. And I'll just repeat that, to provide experimental ASO treatments to these patients for free for life. Operationally, we define the ultra-rare mutation as a mutation that affects between 1 and 30 patients worldwide, and so a very small patient population. However, the scale of the problem is really quite substantial. So it is certainly the case that each of these patients and each of these cases may be very unique. But already millions of this type of patient have been identified. And as more and more genomes of more and more human beings are sequenced, then of course we're going to identify many more of these patients. And one of the items that we're learning that is particularly interesting is that even for well-defined rare diseases that are genetically caused, within that patient population, there are often numerous patients who have unique mutations that are absolutely unique to them. And it is also clear that over the last few years, a number of institutions have focused on this tremendously underserved patient population, and a lot of progress that has been made in diagnosing and genetically characterizing these patients. But in the end, 9 times out of 10, what the patients have to hear is there's no medicine to treat you, and it's highly unlikely that there will be. And so n-Lorem is filling that void. And to date, n-Lorem is the only solution to actually taking a genetic diagnosis and turning it into hope and to treatment. To give you an example of what some of these cases are like and what these parents and patients go through, I'll tell you about a child that we are in the process of treating. These are young parents, and they had a healthy young baby girl as their first child, and of course they were overjoyed. But within a few months, they became concerned that their daughter was not meeting developmental and growth milestones, visited their pediatrician, and had their anxieties allayed that these developmental milestones are quite variable. But over months, it became clear that their child was not advancing in size or developmental milestones. And what followed next is that they spent the next 2 years bouncing from one specialist to the next. Ultimately, their daughter required hospitalization again. And during that hospitalization, this 3-year-old child had 2 cardiac arrests. Because the hospital wasn't equipped to deal with cardiac arrests in a tiny 3-year-old child, she was transferred to a tertiary care center. And there, the diagnosis was promptly made as Cockayne syndrome. And here's one of the real tragedies of these patients. Cockayne syndrome was first described in 1936, and it's been rediscovered enough times that there are other names attached to it. And so it's not a new syndrome. It's actually a known syndrome. And of course, these parents would have to be told, if there were no n-Lorem, that there was no treatment. How to describe this child to you? Well, she's a tiny, tiny, smiling, happy 3-year-old in a stroller because she can't walk. And she has the wizened face and cardiovascular system of a person my age, because she has a rapid aging syndrome. When the parents were asked what their aspirations for n-Lorem for their child were, the mother very quickly responded and said, time, just a little more time. The father thought a little longer and responded with one word, hope. You really need to meet just one of these desperate families to understand how important it is that n-Lorem be successful in meeting the needs of these patients. There is no hope for these patients and their parents and their families. And we already know it can work. In parallel, while I was setting up n-Lorem, we've had the opportunity to help 2 investigators, Tim Yu at Harvard and Neil Schneider at Columbia, actually provide experimental ASO treatments to 9 patients now. And focusing on the FUS mutation very quickly, it's an interesting situation. Here, of course, ALS is a well-known, well-understood disease. And the FUS mutation is unique in that it produces a very aggressive ALS. Typically from symptom onset to death is around 6 months. And in fact, the first patient that we tried to bring an ASO to was already very advanced and requiring respiratory assistance. And so we were very worried that we would not be able to get the ASO to her in time to save her. But we did. And she just recently succumbed to her disease, but she lived a year being treated with the experimental ASO. And so it is clear we can do this, and it's clear that in at least some cases, there can be significant benefit. To look a little more closely at n-Lorem itself, the simplest way to think of this is we're middlemen. We connect the patient or the need, the patient, the investigator, the institution that is taking care of that patient, to the engine for ASO discovery and development that we built at Ionis. And specifically then, we provide a site to which the patients and investigative physicians can apply for treatment, a process that assures quality evaluation of each application and quality preparation of the ASO. We fund then Ionis and our CROs to discover, develop, and get the ASO ready for the treatment of the patient. And then it's really important, I think, that we are absolutely committed to thoughtful evaluation of the performance of each ASO, as well as the overall performance of the entire portfolio. And I think that's a critical component of what we do. Now, to look at it just a little more detail, and then I'm going to get into more detail in the next a couple of slides. The applications comes in, they are blinded so that we're fully HIPAA compliant. They're triaged by Frank Bennett, the CTO. And then if the patient is appropriate to treat with an ASO, that case is presented in more detail to the what we call the Access to Treatment Committee. And this is a critical component of quality decision making. This is a group of individuals that comprise all the various expertise, types of expertise that we need to assure that each of these complex risk-benefit decisions is made thoughtfully. And again, I think that's a critical component of what needs to be in place to do this right. I want to spend another minute or two looking at the processes at n-Lorem in a little more detail, because I think it will give you a better sense of the scope and of activities, and also how we've decided to lay out the various responsibilities. Obviously, submission is the responsibility of the investigative physician and the patient or parent, and it takes both because we require a great deal of information. And then as the application is processed, the triage is done by the CTO of n-Lorem, and sometimes that includes me. If the case is obviously not a candidate for ASO treatment, a brief summary is presented to the ATTC, but if the case is a potential case to treat, significant detail presentations are made, and typically the investigator physician is also involved in that. Now, of course, we turn to n-Lorem, turn to Ionis, and since we invented this, I don't think there's an organization that's more capable of doing this the right way than Ionis. And it is important that it be done the right way, as this group certainly knows. The identification of the optimal site and the optimal design is actually a complicated affair, and it needs to be done by people who know something. And then finally, or next, we work with the investigator closely to help him put an investigator-initiated IND together and file it. And then in the evaluation period, we play critical roles. During about a year that it takes to prepare the ASO, we ask that the investigator and the patient or parent thoroughly document the natural history of their disease. Before we even begin, we define with the investigator a primary treatment endpoint and secondary treatment endpoints. And then during that year, while we're waiting for the ASO to be prepared and the IND filed, the investigator is expected to document the primary and secondary events that we've agreed on, or secondary goals, and the measures that we've agreed on. And so we have a 1 year recent natural history to which we can compare the treatment period. And I think that's critical. And we also plan then to publish all that, and we will also have an annual meeting of investigators in which our learnings can be shared more broadly. And as this expands, of course, that meeting is going to become larger and larger. These are all critical components of quality. And I'll come back again to quality. Quality processes, quality ASO, quality evaluation are critical if this is to be done properly. So I want to just step back and walk you through the journey I made to n-Lorem, because I think it will help you understand a good many things and answer many of your questions. To do this, you need at least 4 components, in my view. You need a technology that makes it feasible. For me, it was important to decide whether a traditional commercial model could work, because that's the model we have experience with, or whether we would have to strike out in new directions that have never been tried before. You need access to the front end, of course, and we need a regulatory environment that is supportive of trying to find solutions for these patients. The technology, of course, it was very obvious to me the intrinsic efficiency of antisense. That's one of the reasons I founded Ionis long ago. But it's more than that you need. Today's antisense, especially at Ionis, is highly versatile. We know how to design ASOs that take advantage of multiple post-binding RNA binding mechanisms. We validated that we can give these agents through essentially all routes of administration. And we understand their PK/PD, and in each of the major organs that we work, we understand the suborgan pharmacokinetics and pharmacodynamics in detail, and that's critical. The automation that Ionis, I think, is unique, and it's informed by 30 years of experience and artificial intelligence contributed algorithms that help us avoid problem sequences and other items. In addition, it's essential that antisense agents are consistent within each chemical class. As you know, within a chemical class, they all have similar potency. They require dosing at about the similar frequency. And Ionis uniquely has over the years put together databases that integrate all of the adverse event observations from non-human primates through all controlled clinical trials for all the chemical classes and routes of administration that we use, and we've published those. And those are available to regulatory agencies as well. So all of that information leads to more effective decision making. And of course, the potency and durability of modern ASOs play a big role in making this feasible. Just to remind you, of course, we have validated that we can administer PSASOs through all routes of administration, including oral. Certainly oral is not a candidate for these patients typically, but it has been shown. And we've created, by learning, a good many new mechanisms that we can use to either downregulate protein or upregulate the translation of specific proteins. And here's just 6 of the mechanisms that we can use and to design ASOs to cause reduction of the protein and or the RNA. And more importantly, we've added to our ability to be agonist-like. In the past, what we had available to us was to alter splicing if the disorder was a splicing disorder. Now, having taken advantage of cis-acting elements that have been discovered in messenger RNAs that downregulate translation of proteins, we have the opportunity to take other approaches to actually behave as an agonist. And in fact, we are developing a UARF ASO for one of these n-Lorem patients. Now focusing on the organs on which we are focused is this slide. Currently, we focus on the central nervous system, liver, lung, kidney, and eye. Why these organs? Because they're the organs we know best. We really understand these organs, and they're also the organs where we get very high potency. Routes of administration in the center column there. Once again, if we can dose locally, that just increases the safety index. But we do have 2 systemic organs that we treat, liver and kidney subcutaneously. And over on the right is the total annual dose. And as you know, about the smallest quantity we can make today under GMP conditions is about 10 grams. And given the stability of these molecules as dry powders, we can use that 10 grams. Certainly that's a 10 or 20 year supply for most of these patients. So it turns out that the cost of the preparation of the ASO is a modest component of the cost structure. The logic for a charitable solution, I think, is very obvious and just begins with the numbers. How does one gain approval for an agent that can treat 1 patient? And even if there are 10 or 20 patients identified worldwide, even managing a clinical trial will be almost impossible because these patients are spread around the world. And so it is very clear that a charitable solution is required. Even if one could get approval, given all of the challenges here, one would have to charge many millions of dollars a year to patients. And I think that's wrong. And I think it would be bad for our industry. On the other hand, n-Lorem is a great opportunity to do something good for these patients and families and to show the true heart of our industry, because a heart is a good heart. Then the front end took a while to be sure that we would have sufficient access to the front end, because we need a lot. We obviously need a diagnosis, but we need a full genomic workup. And typically that's full sequencing, not just exome sequences. We need to understand the organs affected. We need to work with the investigator and the patient or parent to assure that we have a primary treatment goal, as well as our secondary treatment goals that we'd like to see bring benefit. We need an investigator capable of managing an investigator-initiated IND and managing that patient and managing an ASO treatment. And we need an institution willing to participate in all this. That's a lot. Fortunately, I was introduced to the Undiagnosed Disease Network, This is a consortium of tertiary care centers funded by the NIH, who for the last few years have made it their business to diagnose undiagnosable patients and to fully characterize them genetically. And then, since we've been founded, we've interacted with a large number of other institutions. In fact, in our first year, more than half of the applications to treat came from institutions that are not members of the UDN. And that just it testifies to the fact that many institutions see these patients as underserved and are busy trying to understand these patients and at least give them a diagnosis and a genetic cause. So that's great. So we have access. And finally, regulatory. I spent most of 2019 just confirming that the FDA was interested in these patients and was supportive. And I am absolutely pleased that the FDA has been highly interactive and clearly committed to finding solutions for these patients. I was very pleased that the FDA provided initial guidance. It's, of course, just administrative today, but it was important. It was positive. It said this has to be a collaborative effort, and it focused on ASOs. And so we've had quite a bit of input already, and we'll be contributing additional input because our experience, I think, is on point. So we look forward to more definitive guidance, and we certainly don't expect to get everything that we suggested, but we do believe that we will get sufficient guidance that will make this work even easier. And so we're looking forward to that. So all of that then in place, the founding donors were Lonis Pharmaceuticals, our partner Biogen in the neurosciences, and my wife and myself. To date, Lonis has contributed $2.8 million plus more than 70 volunteers have contributed. Biogen has contributed a similar amount, but they'd rather us not give the specific number of dollars. And my wife and I have contributed $3.2 million. And so that got us off to a start. And we launched then in January of last year, and I would say that the launch was quite successful. I think the coverage was very good. Of course, our parallel experience with Tim Yu and Neil Schneider was an important positive experience that provided a lot of information to us. We were well covered in the trade and scientific press, but not covered in the lay press. And I think that was, all the other things going on in 2020 that did that. We've created a large number of collaborations, and that has helped us reduce the cost per patient even more. We had not planned on taking any new donations in 2020, but we have, and I'll go through some of this progress in more detail. One of the most important steps that we've taken is the development of quality systems. And I come back and emphasize the importance of quality systems and quality ASOs, because I am concerned about the number of amateurs out making ASOs and putting them into patients. These people do not seem to understand that ASOs are complex chemicals with pharmacological activity and need to be developed properly. So quality systems that assure appropriate risk-benefit decisions, quality systems that assure the best ASO possible is provided to the patient, quality manufacturing and controls, and then quality methods to assure that the performance of these ASOs is evaluated in a critical fashion, including the publication of those results annually. Always being the most optimistic, I felt in our first year, given the novelty of all this and COVID-19, we might get 5 submissions and maybe be able to treat 1. In fact, we received 54. And we're moving toward treating about 20 of these patients. I do expect that number to go down a bit because clearly we have limitations as the number of applications increase and the diversity of applications increase. And I do believe that in 2021, we'll see a more than arithmetic increase in applications and hopefully not a logarithmic increase because we're already stretched. To give you an example of the complexity of these cases, this is just a sampling of some of the cases that we're proceeding to treat. And the only point that I want to make is these patients come in all ages and descriptions, and typically they have, of course, multiple symptoms and signs of their genetic disease. And so selecting a primary treatment goal and secondary treatment goals and measuring them appropriately, I think, is a critical component. And as I said, we're committed to collaborating. We believe that only through collaborations can the most patients be treated in the best way. And we think collaborations have already enhanced access to these ASOs. We're confident that they will enhance the quality of care, reduce the cost per patient, reduce the time from application to treatment. These patients are desperate. Every minute counts. And enhance our ability to assure that we're doing more good than not. And so we work very hard to establish collaborations. We have a formal collaboration with the UDN. We have formal and informal collaborations with a number of tertiary care institutions. Our CROs, Covance, and then our neighbor Illumina help us with sequencing. And we do have to do quite a bit of sequencing because often these patients are just exome sequenced. Covance and Charles Rivers Laboratory are broadly supporting us. These are our tox CROs, of course. And all of our contract reorganizations that provide toxicology studies are doing that at very steep discounts and providing other kinds of support. And since that's the major cost center here, that's had a significant impact. And we've already reduced the cost per patient by about 40%. I am in conversations now with our contract manufacturers, and I'm optimistic based on those conversations that they'll become collaborators as well. The more collaborators we have, the more good we can do, the more patients we can help. And we've proposed even that if the policies allow, the FDA participate in this with us. Another big step that we've taken is to just simply broaden the awareness. And we have a number of physician communities that need to be brought to be aware of n-Lorem. We've made good progress in some areas, and we have a lot of work left to do in 2020 in some of the other areas. We've also spent a great deal of time just going door to door, speaking to various institutions, and that's had a positive effect. We're getting applications from many of these institutions. We've spoken to a number of patient advocacy groups. I've introduced n-Lorem to pharma, bio, and as of today, to OTS. And I've spoken to a good number of pharma and biotech companies, inviting them to join us, set up their own organization and liaise with us. It really doesn't matter how that gets done. What matters is it's done well and that these patients are given hope in treatment. We have not formally started fundraising yet. I didn't want to raise money formally until I was sure this could work. We're just now beginning to put the materials together to start formal fundraising. We have still not succeeded in getting the lay media to cover us, but we're making progress there, and I'm confident it will happen in 21. So we have more work to do, but I'm very pleased with the progress we made. We have developed novel methods of evaluating the performance of ASOs in single patients. It turns out that there is a literature on N of 1 studies, but almost all of them are crossover designs, which won't work in these patients because they are desperate, and our drugs are very long duration. And so the process that we put together is very patient-centric, and it's focused on clinical evaluations that are validated And it depends on then that year of pre-treatment, thorough natural history with a focus on defining the primary treatment goal and the secondary treatment goals, measure them thoroughly the year before treatment begins, and then measure them carefully after treatment begins, publish those results, and share those results every year with the investigators via an investigator in the meeting. And even though we had not planned to take additional donations, numerous donors have stepped up. Here's a list of some of the donors that we've acquired, and this added several million more dollars to us. And so it gives me optimism that now that we're getting after this formally, that we'll be able to raise sufficient funds to meet the needs of these patients. Obviously, the technology is scalable, and the problem is enormous. And so my greatest fear is that someday I'll have to tell a patient that we can't treat them because we don't have sufficient funds. But given the overwhelmingly positive response to date, I'm optimistic that we're going to be able to keep pace with the demand. So that's n-Lorem. I've been asked what my long-term vision for n-Lorem is. And my answer is I don't have a big dream for n-Lorem. It's a very tiny dream. My dream for n-Lorem is that we help a single patient in a single family, one family at a time. And I think if we do that, that will change the world too. It certainly will change the future of not just the patient, but these families. And I'm speaking to OTS to hope that OTS, the society that represents the companies that created the technology that makes n-Lorem possible, will become an active contributor and participant. I can assure you that it is a worthwhile and incredibly individually, personally rewarding experience to be doing this. Thank you for the opportunity to talk to you, and now I'll answer questions. Give me just a minute here. The first question is a scientific question, my opinion on phosphorothioate chirality. I don't want to spend any time on that, but we published a thorough paper in NER a year or so ago. I think it's Ostergaard is the first author. So have a look at that paper. That doesn't cover my opinion. It covers the data and the conclusions that derive from it. The next question is germane to n-Lorem. We are currently, and the question has to do with countries outside the US. And the specific focus was on developing countries. I think that is a challenge that we're going to have to wait to try to meet. Today, n-Lorem is limited to the United States. And the reason is simple. The thought of trying to deal with multiple regulatory agencies with something as novel as n-Lorem was daunting. Not only regulatory agencies, but also payers and health authorities and so on. So my plan is to get n-Lorem situated and successful in the United States, achieve effective guidance from the FDA, and then begin to export n-Lorem to the developed world. I think the challenge in the developing world is really quite significant, because to manage these patients, you need care facilities that simply don't exist in the developing world. And so I think that remains to be conquered in due course. And I think the first step to conquering it is making them developing countries, but rather developed. And once again, that's I was asked that question twice. n-Lorem is accepting today the question of what cases are we accepting. We are accepting cases where the primary treatment need is in those organs that I described: the central nervous system, the liver, the kidney, the eye, and the lung. And as we learn more about these agents in other organs and we improve the potency, then when I feel that we have sufficient information, sufficient potency to expose these patients to only prudent risk, then we'll move to those organs. I think it is really important to be cautious with these patients. They are desperate. And the last thing we want to do is add unnecessary challenges for these patients because of side effects that we could have avoided. The next question is a process for handling reapplications. We don't think so. Let me give you the philosophic answer, which I've tried to teach. We do this one patient, one mutation at a time, and the bias is to treat. So if there's a case in which we have insufficient information, we let the treating the applicant know the reasons we can't treat. If it's a set of things that we might be able to figure out ourselves by doing some additional work, we now have sufficient funds and support that we go ahead and do that. If it requires more work, then we look forward to getting the application back when there's sufficient information for us to make a decision. One mutation, one patient at a time. Another point of confusion for people is, what diseases do we focus on? Well, if you think about it, once you know the genetic cause, the name of the disease is irrelevant. So we're agnostic as to the name of the disease. We're driven by the nature of the mutation. And is it a mutation that we can address? Is it in an organ that we're comfortable treating? And is there a primary treatment goal that we think we can measure that would be meaningful to the patient? One patient, one mutation, one goal at a time. The next question had to do with guidelines for chemistries. Well, I think the only chemistries I'd use are the chemistries we've invented and we've studied. I think anything else is not understood well enough. And so in these patients, we're using 2' methoxys when appropriate. We're using 2' CETs when appropriate. We're using all the different designs that we have, and those designs continue to evolve. And we're using ASOs with targeting ligands like GalNAc or GLP-1. And as the technology evolves, then we'll add new targeting ligands and new designs. So I think today, if someone wants to treat these patients with an ASO, they have to talk to n-Lorem. Again, another question on countries. We do hope to move very rapidly to Canada, but I think our first step is to get thorough guidance out of the US. And then our next step is the developed worlds. We have already gotten applications from countries outside the US, and it's heartbreaking to have to say no. But if we don't do this carefully and we don't do this right, and we don't tackle one thing that we can tackle at a time and do it right, I fear the whole thing could fail. And I think that's a big risk that I'm trying to avoid. I hope you understand. The question about model systems, I think that's an important point for me to clarify. The work that we do preclinically is very limited. Typically, what do we need to know? We need to know the nature of the mutation. Is it something we can address? We need to know the function of that gene so that we know what the on-target risk of altering it is. And we can do that in cell systems that we have. But we're doing it more and more in patient-derived cells because in many cases, these patients have already been worked up and their cells are available. And then we do a toxicity screen to identify any, the very, very best of the group of ASOs that we've designed and looked at. And then we move to a single species tox study that typically is about 3 months. And armed with that and the GMP for synthesis and analytical data, we then begin treating the patients. Quality metrics for ASO development. Well, I think you need access to, first, all the automation that we have and the algorithms that we have. We typically will screen 1,000 or more sites in an RNA with multiple different designs and chemistries and so on, depending on what our goal is. Out of that, and that's done very rapidly and inexpensively. Out of that, then we identify often, a good number of ASOs that look very good. We do then in vitro full dose response curves and the like. And then typically we will look in the patient-derived cells. If there is an appropriate animal model, then we'll consider that for these patients, but that's rarely the case. And it's not really necessary. And so it is those quality systems that I think you have to go through, which are basically the same quality systems that we use to make commercial ASOs, just limited in the total scope of activity and the total amount of work that we do. And then obviously we work with CMOs that are organizations that we've helped make sure that they have all the right synthetic schemes and quality systems and so on. And we're very confident in working with them. And, there are now a large number of those organizations. So all that's available. I think equally, maybe even more important, is a commitment to quality evaluation of the performance of these ASOs in the clinic and disseminating that information broadly to investigators and to the scientific community. And I really like the systems that we've put together to do that. I think I think these are the right ways to go. Yes. I have another question, once again, about outside the US. I regret to say that we're not able yet to treat a patient in Europe. We have had patients who had the means to move, that is, parents to move, but we really at the moment are limited to institutions in the United States. I'm hopeful that by the end of 21, we'll be geared up with guidance from the FDA to begin then the conversations with the regulatory agencies in other countries. But it's not just the regulatory agencies, it's also the health agencies and what they're willing to do for these patients. And so I think that's a complicated process. And I feel very, very committed to one step at a time. We are up and walking. We're not up and running. And what we cannot do is stumble. If we stumble and our aspirations are unmet, then there are going to be patients who die. and who might not have had to. So one step at a time, and caution, I think, is the right approach. I think I've covered all the questions. Once again, I thank you for your interest, and I look forward to opportunities to work closely with OTS to expand the effort and to be fully successful at helping these poor families. They need our help. Thank you.
Thank you very much for doing this really inspiring talk for OTS, and I hope we can help publicize this effort a bit more for you. If there are any more questions, I think there's one last one, and I think the end of that question about the treatment was more about how likely are n-Lorem's individualized solutions to facilitate treatments for other patients. The end of that big long question.
That's a good question. It is certainly the case that what looks like an N of 1 can sometimes turn out to be 10 or 20 or more patients. Obviously, if we get a second application with the exact same mutation, we can use the same oligo. And so we look forward to getting those because it's a great way to leverage, the work that we've done for that first patient. So far, we haven't had a case other than the FUS mutations with exactly the same mutation, but I'm sure we're going to get it, and that'd be great if we do.
Yeah, because then you can start treatment immediately with the already available oligo. So
we could, and it's of course less expensive. It would spread the cost across a number of patients, and we might. I mean, I would, since we haven't dealt with it yet, and every step we take is brand new. We would really look carefully at it and make sure that there wasn't additional work that needed to be done. But in the simplest case, we just say, yes, we have an ASO on the shelf and we can begin treating you very quickly. And since we would already have an investigator IND filed, that even the regulatory process should be facilitated. So I'm looking forward to getting those applications that have the same mutation. They'll be nice and easy for us to manage.
Yeah, I think I'd also like to mention that at least here in the UK, I think there is some effort to help with this kind of treatment with NATA. And I believe in the Netherlands as well, Dr. Annemieke Aertsma-Roos is leading that there, I think.
Yes, and there may be organizations in those countries that set up to do this. I just hope that they work with us, that to take advantage of the 30 plus years of knowledge that we've accumulated at Ionis and the processes and whatnot that we've set up at n-Lorem, that I think if someone has a better idea and they come up with a system that works even better, great. But we've done a lot of thinking. I've done a lot of thinking about this, and that thinking is represented here. And the experience that we've gained is valuable, and I hope people take advantage of it.
Yes. Thank you very much again. Yes, the recording will be available as usual on the same webpage that you signed up on, and also from the OTS YouTube channel. But please wait for a day or two before you look. Thank you, and I wish you a good day.
Thanks very much, everyone.
It's been an honor.